Reactions of monosaccharides. Carbohydrates are hydrophilic substances with potential or free aldehyde or keto group and a number of hydroxyl groups. Monosaccharides, the basic components in the series, have a single carbon chain glucose like analdohexose and fructose, hexoketose, and all the most common monosaccharides. Perform the test with solution of glucose and fructose. First, we will be conducting Modish test. This is a general test for all carbohydrates. Carbohydrates, when heated with concentrated H2SO4, undergo dehydration to give a furfural derivative. Hexose, for example, form hydroxymethyl furfural. This compounds condense with alpha naphthol to form a colored product. To 1 ml of glucose solution, add 2 to 3 drops of 1% alpha naphthol. Mix and add it carefully through the sides of the test tube. 2 ml of concentrated H2SO4 is added so as to form 2 layers. Note the formation of a reddish violet ring at the junction of the 2 liquids. Repeat the test with fructose. Hydroxymethyl furfural chromogen is formed. Benedict test. Reducing sugars under alkaline condition tautomerize to form any diols and enediols are unstable and decompose to yield a variety of products. 1,2 enediols will give formaldehyde and pentose. The chain reaction continues to produce short chain aldehyde, which are powerful reducing agents. They can reduce the cupric ion to cuprous ion, which is the basis of Benedict's reaction. Which is the base of Benedict's reaction. The cuprous hydroxide formed is not only soluble is not easily soluble. In order to keep the hydroxide in solution, a metal chelate like a citrate tartarate is introduced into the solution. Benedict's solution contains the milder alkali Na2CO3. When reducing sugar solution is treated with Benedict's reagent, the sugar is decomposed and the product reduces cupric ions to cuprous ions as yellow cuprous hydroxide. Sugar Benedict sugar acid. Cuprous hydroxide during the process of heating is converted into red cuprous oxide. The Benedict reagent test is more specific than Felling's test. To 2 ml of glucose solution, add 3 ml of Benedict's reagent. Mix and boil 4 minutes over a small flame. Note the formation of red precipitate. Repeat with fructose. And Buffett's test. Aldose and ketosis can reduce cupric ions even in acidic conditions. Monosaccharides react very fast, whereas the reaction with reducing monosaccharides is slow. This property is used in Buffett's test to distinguish a reducing monosaccharide from a reducing disaccharide. The Cu2 plus formed is treated with phosphomolybdic acid. The colorless phosphomolybdic acid is reduced to blue phosphomolybdic acid. Perform the test simultaneously with glucose and fructose. To 2 ml each of the fructose and glucose solution is taken in test tube and add 2 ml of Buffett's reagent to each. Keep the test tube 3 minutes in a boiling water bath. Cool under running water. Add phosphomolybdic acid drop by drop with mixing till the solution is clear. A deep blue color is formed in both the instances. Then Celevano's test. Composition of Celevano's reagent. Resorcinol in dilute HCl. Principle. HCl is a Celevano's reagent. Dehydrates the ketosis to form furfural derivatives, which condenses with resorcinol to form cherry red color complex. This test is used to distinguish between aldoses and ketoses. Prolonged boiling may also give a positive Celevano's test for aldose due to its conversion in keto group by HCl. Then Nosazone test. Principle. And reducing sugars are treated with the phenylhydrazine. First, phenylhydrazones soluble are formed. On heating, these hydrazones further react with 2 molecules of phenylhydrazine to form sugar osazone, which is insoluble. Non-reducing sugars like sucrose does not form osazone. Okay, this is a structure which represents glucosazone or fructosazone. Similar structure for them. Glucose plus phenylhydrazine, glucose phenylhydrazine, deoxy sugar, glucosazone, phenylhydrazine. 4. Distinguishing the different reducing sugars in urine, etc. under the conditions of glucosuria or lactosuria. This is the only test to differentiate between maltose and lactose. Then only reducing sugars which have a free aldehyde or keto group will form osazone crystals. Glucose, fructose and mannose form identical osazones. So this is because the sugars differ only at carbon number 1 and 2. Osazone formation involves C1 and C2. So the difference between these sugars at C1 and C2 are masked during osazone formation. Then glucosazone and fructosazone crystals are formed during boiling itself. Then maltosazone and lactosazone crystals are not formed during the process of heating, boiling, because osazone of these sugars are soluble in hot solution. Hence, osazones of maltose and lactose are found only on cooling. Glucose, fructose and mannose gives a positive test for osazone reaction. All of them form colored needle-shaped crystals since they are highly insoluble. The crystals are named glucosazone, fructosazone and mannosazone respectively. So remember, while performing this osazone test, glucose and fructose will develop that crystals earlier before cooling. But in case of maltose, that will develop only after cooling. Does not form on heating. Okay. So reactions of glucose experiment, solubility test. First, we are going to learn about reactions of glucose. Solubility test. Test the solubility of given substance in water. Observation. Experiment. Solubility test. Test the solubility of given substance in water. Observation. Soluble. Inference. Absence of polysaccharide. Then Molish test. Take 1 ml of test solution in a clean and dry test tube. Add 1 to 2 drops of Molish reagent. Mix well and add 1 to 2 ml of concentrated H2SO4 along the sides of the test tube without shaking. A reddish violet ring at the junction of two liquids. Presence of carbohydrates. They are dehydrated by concentrated H2SO4 to form furfural or hydroxymethyl furfural. This condenses with alpha naphthol to give a violet ring at the junction of the two liquids. Fillings test. To 1 ml of test solution, add 1 ml of filling solution A and B and mix well. Heat in boiling water. The bath for 3 minutes. Reddish brown precipitate formed. Presence of reducing sugar in alkaline medium. Reddish brown precipitate formed. Presence of reducing sugar in alkaline medium. Carbohydrate with free aldehyde or ketone group reduces the cupric ion to cuprous ion. Reducing sugar. Remember, reducing sugar. Fillings test positive indicate reducing sugar. Reducing sugar. Reducing sugar. Okay. Here you could see a structure of this glucose is on. Then the cupric hydroxide formed is concentrated to cuprous oxide, which gives reddish brown precipitate on heating. Then Benedict's test. Take 5 ml of Benedict's reagent in a clean dry test tube and add 8 drops of test solution. Boil it for exactly 2 minutes. Brick red precipitate. Presence of monosaccharide in the presence of an acid medium. Presence of monosaccharide in the presence of an acid medium. The carbohydrate with free aldehyde or ketone groups reduces the cuprous ion to cupric ion. The cuprous hydroxide this formed is converted to cuprous oxide, which is red scum on heating. Benedict's test. Here, one thing to remember, we need to take first the Benedict's reagent. Then after that, we will be taking the solution. Don't do this in reverse. Okay. Then next osazone test. Take 3 ml of test solution in a clean dry test tube. 3 ml of test solution in a clean dry test tube. Add 1 spatula of phenyl hydrazine hydrochloride, an equal amount of sodium acetate, and 2 to 3 drops of glacial acetic acid. Mix well and keep the test tube in boiling water bath for 20 minutes. Note the formation of yellow crystals. Allow the test tube to cool under tap water. Take out the crystals with the help of a glass rod. Mount on a glass slide using cover slip and observe under the microscope. Once again, take 3 ml of the test solution in a clean dry test tube. Add 1 spatula of phenyl hydrazine hydrochloride, an equal amount of sodium acetate, then glacial acetic acid, 1 to 2 drops. Equal amount of phenyl hydrazine hydrochloride and equal amount of sodium acetate. and 1 to 2 drops of glacial acetic acid. Mix well and keep the test tube in boiling water bath for 20 minutes. Note the formation of yellow crystals. Allow the test tube to cool under tap water. Take out the crystals with the help of a glass rod. Mount on glass slide using cover slip and observe under the microscope. Okay. You could see needle or broomstick shaped crystals. Presence of glucose. Glucose reacts with phenylhydrazine sodium acetate mixture 1:1 to form crystalline glucosazone. Next, Benedict test. Take 5 ml of Benedict's reagent in a clean dry test tube and add 8 drops of test solution. Boil it for exactly 2 minutes. Brick red precipitate. Presence of monosaccharide. In the presence of an acid medium, the carbohydrate with free aldehyde or ketone groups reduces the cuprous ion to cupric ion. The cuprous hydroxide thus formed is converted to cuprous oxide. This is red scum on heating. Cu2 plus Cu plus COH Cu2O, red color. Cu2 plus turns to Cu plus COH Cu2O. Ozone test. Take 3 ml of test solution in a clean dry test tube and add 1 spatula of phenylhydrazine hydrochloride and equal amount of sodium acetate 1:1 ratio and 2 to 3 drops of glacial acetic acid. Mix well and keep the test tube in boiling water bath for 20 minutes. Note the formation of yellow crystals. Allow the test tube to cool under tap water. Take out the crystals with the help of glass rod. Mount on a glass slide using cover slip and observe under the microscope. You could see needle-shaped crystals, presence of glucose. We just learned this. Next, reactions of fructose. To a greater extent, it is similar with the glucose, but still we are going to learn this. First, we will be performing solubility test. Test the solubility of the given substance in water soluble. So that indicate absence of polysaccharide. Then Molish test. Take 1 ml of test solution in a clean dry test tube and add 2 to 3 drops of Molish reagent. Mix well and add 1 to 2 ml of concentrated H2SO4 along the sides of the test tube without shaking. A reddish brown ring at the junction of two liquids. Presence of that will indicate a reddish brown ring at the junction of the two liquids. Presence of carbohydrate. They are dehydrated by concentrated H2SO4 to form furfural or hydroxymethyl furfural. This condenses with alpha and naphthol to form violet color at the junction of two liquids. Then Felling's test. To 1 ml of test solution, add 1 ml of each filling solution A and B. Mix well and heat on boiling water bath for 3 minutes. Reddish brown precipitate is formed in the presence of reducing sugar. In the presence of alkaline medium, the carbohydrate with free aldehyde or keto group reduces cupric ion to cuprous ion. The cuprous hydroxide formed is converted to cuprous ion, which gives reddish brown precipitate on heating. Cu2 plus Cu plus COH Cu2O. Baphoit's test. This is a new test. Take 1 ml of test solution in a clean dry test tube. Add 2 ml of Baphoit's reagent. Mix well and boil it for 1 minute. Red scum is formed. Presence of monosaccharide. In acidic medium, the carbohydrate with free aldehyde or keto group reduces the cuprous to cupric ion. The cuprous hydroxide thus formed is converted to cuprous oxide, which gives red scum on heating. Cu2 plus Cu plus COH Cu2O. Celavano's test. Take 3 ml of Celavano's reagent in a clean dry test tube and add 1 ml of test solution. Mix and boil for 30 seconds. Cool the test tube and observe. Cherry red color. Presence of fructose. Remember, Celavano's test is for fructose. Okay. Earlier we mentioned about Baphoit's test that create a red scum. Okay. Then Celavano's test. Take Celavano's reagent, add the solution into it and heat it. Okay. That will create cherry red color. Presence of fructose. In acidic medium, ketones on reaction with acids give furfural derivatives which further react with resorcinol to form red colored complex. Presence of fructose. In acidic medium, ketones on reaction with acids gives furfural derivatives which further react with resorcinol to form red colored complex. Ketones, resorcinol colored complex. And Folger's test. to 1 ml of test solution, add 3 ml of Folger's reagent. Heat it in boiling water bath for 3 minutes and cool. What is the result? Blue color is obtained. Presence of fructose. Ketose on reaction with acid gives furfural derivative. They condense with Folger's reagent to form a colored complex. Blue color. Just remember this one. You need to add that Folger's reagent and heat. That's it. Then blue color is obtained. Presence of fructose. Ketose on reaction with acid gives furfural derivative. They condense with Folger's reaction to form a colored complex. Ketose, acid, Folger's reagent, colored complex. Then analysis on test. Take a 3 ml of test solution in a clean dry test tube. Add 1 spatula of phenyl hydrazine hydrochloride and add 2 to 3 drops of glacial acetic acid. Then before that, sodium acetate is also added. Then mix well and keep the test tube in boiling water for 20 minutes. Note the formation of yellow crystals. Allow the test tube to cool under tap water. Take out the crystals with the help of a glass rod. Mount on a glass slide using the cover slip and observe under microscope. You could see yellow thin needle from stick-shaped crystals. Presence of fructose. Fructose reacts with phenyl hydrazine sodium acetate mixture to form fructose. So remember, Sylvanus test we just exclusively perform for fructose. Sylvanus test. Take Sylvanus reagent 3 ml and add the test solution of fructose. Boil. You could see cherry red color. That indicate presence of fructose. Okay. Then a Folger's test is performed if we need to do that. We won't be performing that, but still we will be learning about it. Okay, then fructose is formed. Reactions of disaccharides. Disaccharides are formed by the union of two monosaccharide units by glycosidic linkage with the elimination of one molecule of water. It may be reducing or non-reducing depending on the availability of free or potential aldehyde or keto group in their molecular structure. The disaccharides of biological importance are maltose, lactose and sucrose. Maltose, lactose and sucrose. We just learned about glucose and fructose. These are monosaccharides. Later is about disaccharides. For example, maltose, lactose and sucrose. First, we will be performing a Molish test to identify the presence of a carbohydrate. Composition of Molish reagent: 1% alpha naphthol in 95% ethanol. Principle: carbohydrates with concentrated H2SO4 undergo dehydration to give furfural or furfural derivatives. This compounds condense with alpha naphthol to form a reddish violet ring. This is a general test for all carbohydrates. Excess alpha naphthol solution and impurities in the reagent may give a green colored ring. Molish test is given by carbohydrates with at least 5 carbons. Therefore, fructose and trioses and tetroses does not answer this Molish test. This is a general test for all carbohydrates. Excess alpha naphthol solution and this compounds condense with alpha naphthol to form a reddish violet ring. This is general test for all carbohydrates. Excess alpha naphthol solution and impurities in the reagent may give a green colored ring. Molish test is given by carbohydrates with at least 5 carbons. Therefore, trioses and tetroses does not answer this test. So, interaction of acid with water produce heat and can cause chaining of carbohydrates resulting in the formation of a black ring. Therefore, acid should be layered very slowly and carefully to minimize interactions. Then, Benedict's test. Composition of Benedict's reagent: copper sulfate provides cupric ions, sodium acetate provide alkaline medium, sodium citrate provide precipitation of cupric ion. Chelating agent, acts as a chelating agent. Principle: reducing sugars under alkaline conditions form enediols. These enediols are powerful reducing agents and are unstable. Hence, they decompose to yield a mixture of aldehyde that reduce cupric ion to cuprous ion as cuprous hydroxide, COOH. The COOH during the process of heating get converted to different colored cuprous oxide Cu2O precipitate which indicate presence of reducing sugar. 2CuOH twice gives Cu2O plus H2O. Benedict test is also used in semi-quantitative test because of color of the precipitate gives approximate percentage of sugar excreted in urine. This is the test for identification for reducing sugars such as glucose, fructose, maltose and lactose. This test is more sensitive test compared to other reduction tests. Then Buffoyt's test. Composition of Buffoyt's reagent, copper acetate, provides cupric ions. Copper acetate. Copper acetate provides cupric ions. Acetic acid provides acid medium. Principle. It is a reducing test, but it takes place in an acidic medium. In mild acidic medium, reducing sugar undergo tautomerization to form enediols which reduce cuprous ion to cuprous ion. So, cuprous hydroxide informed during the process of heating. Cuprous hydroxide is converted into cuprous oxide which gives red precipitate. So, monosaccharides react first, whereas reaction with disaccharides is low. Therefore, the test is used to differentiate monosaccharides and disaccharides. Higher concentration of disaccharide tend to give a positive Buffoyt's test. Prolonged boiling of for 7 to 12 minutes may give a positive Buffoyt test. Let's come for disaccharides. Benedict test after hydrolysis. Perform this test only for sucrose. The glycosidic linkage is easily hydrolyzed by dilute acid at elevated temperature. The products formed, glucose and fructose, reduce the Benedict's reagent to 1 ml of sucrose solution. Add 5 drops of concentrated HCl. Boil the solution for 2 minutes. Cool and then neutralize. Then hydrolyze it with 5 drops of 40% NaOH. Check using a piece of red litmus paper. Perform Benedict's test with neutralized solution. Here you could see 2 pictures. Lactose is on hedgehog shaped, soluble in hot water, yellow in color, crystallize on cooling. Lactose is on hedgehog shaped. Lactose is on hedgehog shaped, soluble in hot water. So only precipitate out after cooling. So remember that. And yellow in color, crystallize on cooling. Same is the case with maltose is on sunflower shaped. Shape is different, not hedgehog shaped. It's a sunflower shaped one. Then yellow in color, soluble in hot water, crystallizes on cooling. Try to draw this. Osazone formation and mechanism. Glucose, phenylhydrazine, phenylhydrazone. Then alpha keto derivative, distributed glucose. Then enediamine derivative, enoderivative glucosone is formed. Then soluble red precipitate of cuprous oxide is formed earlier. Then Celavano's test. Composition of Celavano's reagent. Resorcinol with dilute HCl. Principle HCl is a Celavano's reagent. Dehydrates the ketosis to form furfural derivatives which condense with resorcinol to form cherry red colored complex. The test is used to distinguish between aldoses and ketosis. Prolonged boiling may also give a positive Celavano's test for aldoses due to their conversion to keto group by HCl. Once again, composition of Celavano's reagent. Resorcinol with the dilute HCl, principal HCl in Celavano's reagent, dehydrates the ketosis to form furfural derivatives which condense with the resorcinol to form cherry red colored complex. This test is used to distinguish between aldoses and ketosis. Prolonged boiling may also give a positive Celavano's result for aldoses due to their conversion to keto group by HCl. Then Osazone test, we know that when reducing sugars are treated with the phenylhydrazine, first phenylhydrazones which are soluble are formed. On heating, these hydrazones further react with 2 molecules of phenylhydrazine to form sugar osagenes insoluble. Non-reducing sugars like sucrose does not form an osazone. For distinguishing the different reducing sugars in urine under conditions of glucosuria and lactosuria, this is only test to differentiate between maltose and lactose. Only reducing sugars which have a free aldehyde or ketone group will form osazone crystals. So glucose, fructose and mannose form identical osazone. This is because these sugars differ only at carbon number 1, 2 and osazone formation involves C1 and C3, C2. So the difference between these sugars at C1 and C2 are masked during osazone formation. So glucosazone and fructosazone crystals are formed during boiling itself. Maltosazone and lactosazone crystals are not formed during the process of heating or boiling because enzymes of these sugars, osazones of these sugars are soluble in hot solution. Hence, osazones of maltose and lactose are formed only on cooling. Reactions of lactose. Remember, lactose is a disaccharide. That's why we are separately dealing with them. Okay. First solubility test. Test the solubility of given substance in water. Soluble, absence of polysaccharide. Then Molish test. Take 1 ml of test solution in a clean dry test tube and add 2 to 3 drops of Molish reagent. Mix it well and add 1 to 2 ml of concentrated H2SO4 along the sides of the test tube without shaking. Take 1 ml of test solution in a clean dry test tube and add 2 to 3 drops of Molish reagent. Mix it well and add 1 to 2 drops of concentrated H2SO4 along the sides of the test tube without shaking. A reddish violet ring at the junction of two liquids. Presence of carbohydrates. It is dehydrated by concentrated H2SO4 to form hydroxymethyl furfural or furfural, which condenses with alpha naphthol to form a violet ring at the junction of the two liquids. Then fillings test. 2 ml of test solution, add 1 ml of each fillings A and fillings B solution and mix it and heat in a boiling water bath for 3 minutes. Reddish brown colored precipitate is obtained. Presence of reducing sugar. In alkaline medium, the carbohydrate with free aldehyde or ketone group reduces the cupric ion to cuprous ion. The cuprous hydroxide formed is converted to cuprous oxide, from which gives red precipitate on heating. So Cu2 plus turns to Cu plus, then CuOH, Cu2O, which is red. Okay. Then Benedict test. Take 5 ml of Benedict's reagent in a clean dry test tube and add 8 drops of test solution and boil it for exactly 2 minutes. A brick red precipitate. Presence of reducing sugars in alkaline medium. The carbohydrate with free aldehyde or ketone group reduces the cuprous ion to cuprous ion. The CuOH formed is converted into the cuprous oxide, from which gives red precipitate on heating. So Cu2 plus sugar OH, Cu plus CuOH, Cu2O red. Nitric acid test. To 1 ml of test solution, add 1 ml of concentrated HNO3. To 1 ml of test solution, add 1 ml of concentrated HNO3 and heat it in a boiling water bath till the brown fumes stop evolving. Keep it overnight and observe the crystals under microscope. Broken glass piece-like crystals are obtained. Presence of lactose. And osazone test. Take 3 ml of test solution in a test tube and add 1 spatula of phenyl hydrazine hydrochloride. Add equal amount of sodium acetate and 2 to 3 drops of glacial acetic acid. Mix well and keep the test tube in a boiling water bath for 20 minutes. Note the formation of yellow crystals. Allow the test tube to cool under tap water. Take out the crystals with the help of a glass roll. Mount on glass slide using cover slip and observe under microscope. And don't forget to spread them. Okay. Then powder puff or hedgehog shaped crystals. Presence of lactose. Lactose react with phenyl hydrazine sodium acetate mixture to form lactose. Reactions of maltose. Solubility test. Test the solubility of given substance in water. Soluble, absence of polysaccharide. Molish test. Take 1 ml of test solution in a clean dry test tube. Add 2 to 3 drops of Molish reagent. Mix it well and add 1 to 2 drops of concentrated H2SO4 along the sides of the test tube without shaking. A reddish violet ring at the junction of two liquids. Presence of carbohydrate. They are dehydrated by concentrated H2SO4 to form furfural or hydroxymethyl furfural. This condenses with alpha naphthol hot and give violet ring at the junction of the two liquids, carbohydrate, alpha naphthol. Fillings test. To 1 ml of test solution, add 1 ml of fillings solution A and B. Mix well. Heat the boiling water for 3 minutes. Reddish brown precipitate is formed. Presence of reducing sugars in alkaline medium. The carbohydrate with free aldehyde or ketone group reduces the cupric ion to cuprous ion. The COH formed is then converted to cuprous oxide, which gives red precipitate on heating. Cu2 plus, Cu plus, COH, Cu2O, red. Ozone test. Take 3 ml of test solution in a clean dry test tube. Add 1 spatula of phenyl hydrazine hydrochloride, an equal amount of sodium acetate and 2 to 3 drops of glacial acetic acid. Mix well and keep the test tube in a boiling water bath for 20 minutes. Note the formation of yellow crystals. Allow the test tube to cool under tap water. Take out crystals with the help of a glass rod. Mount on a glass rod to slide using a cover slip and observe under microscope. We could see sunflower shaped crystals. Presence of maltose. Maltose reacts with phenyl hydrazine sodium acetate mixture to form maltosazone, which confirms the presence of maltose in test tube solution provided. Presence of maltose. Maltose react with phenyl hydrazine sodium acetate mixture to form maltosazone, which confirms the presence of maltose in test tube solution provided. Sunflower shaped crystals. Until now we learned about reactions related to monosaccharide and disaccharide. Next we are moving on to the reactions of polysaccharides. Remember, polysaccharides are also carbohydrates, so Molish test will be positive. Okay, so polysaccharides are the macromolecule polymers of monosaccharides unit linked by glycosidic linkages. On the basis of types of monosaccharide unit present, the polysaccharides are further classified into homopolysaccharides, starch, glycogen, cellulose, chitin, etc. and heteropolysaccharides also there, heparin, hyaluronic acid, keratin sulfate, starch. Starch is a heteropolysaccharide. It is made up of starch occurs in plants, particularly in tubers, roots and seeds and reserve foods. Tubers, roots and seeds as reserve foods. This is made up of amylose and amylopectin. Amylose is a straight chain polymer of glucose molecule which form the helical structure of this water soluble. Then Molish test is performed. This is general test for all carbohydrates. Carbohydrates when treated with concentrated H2SO4 undergo dehydration to give furfural derivatives. Hexoses, for example, forms hydroxymethyl furfural. These compounds condense with alpha naphthol to form colored products. Under acidic conditions, the polysaccharides are hydrolyzed to monosaccharides, which answers the test. Then Benedict test. Reducing sugars in alkaline medium tautomerize to form ene diols, and the 1,2 ene diols will give formaldehyde and pentose. The chain reaction continue to produce short chain aldehydes, which gives powerful reducing agents. They can reduce cupric ion to cuprous ion, which is the basis for Benedict's reaction. The cuprous hydroxide formed is not easily soluble. In order to keep the hydroxide in solution, a metal chelate like citrate in tartarate is included in the solution. Benedict solution contains the milder alkali Na2CO3. When reducing sugar solutions is heated with Benedict's reagent, the sugar is decomposed and reduce the cuprous ion to cuprous ion. Yellow cuprous hydroxide. Cuprous hydroxide produced during the process of heating is converted into red cuprous oxide. 2CuOH gives U2O plus H2O. Iodine reaction. A test specifically meant for starch. So we will be learning about that. So iodine reaction. The amylose component of starch has helical structure. Amylose and amylopectin. Amylose structure. Amylose component of starch has helical structure. When it is treated with iodine solution, iodine is inside the coil, and the complex has an intense blue color. When starch solution is heated, the helical conformation is disrupted and it loses capacity to bind iodine. On cooling, the original conformation is required and capacity to bind is also recovered. Amylopectin and glycogen, because of branching structure, gives light purple and red color with iodine. Benedict test after hydrolysis. Starch and dextrins are hydrolyzed by hot dilute mineral acids to yield glucose, which answers Benedict's reaction. To 1 ml of starch solution, add 5 drops of concentrated HCl. Boil the solution for 2 minutes. Cool it and then neutralize hydrolyze it with 5 drops of 40% NaOH. Check using a piece of red litmus paper. Perform Benedict's test with the neutralized solution. The starch and dextrins give positive test. Reactions of starch experiment. Solubility test. Test the solubility of a given substance in water. Partially soluble. Presence of polysaccharide, not completely soluble. Partially soluble. Presence of polysaccharide. Molish test. Take 1 ml of test solution in a clean and dry test tube and add 2 to 3 drops of Molish reagent. Mix it well and add 1 to 2 ml of concentrated H2SO4 along the sides of the test tube without shaking. A reddish violet ring at the junction of 2 liquids. Presence of carbohydrates. They are dehydrated by concentrated H2SO4 to form furfural derivatives, which condenses with alpha naphthol to form a violet ring at the junction of these 2 liquids. Iodine test. Take 2 ml of test solution and add 2 to 3 drops of iodine solution. Heat and then cool under tap water. Deep blue color on addition of this iodine solution. Blue color disappears on heating and reappears on cooling. Presence of starch. Amylose, component of starch, has helical structure. When it is treated with iodine solution, it is trapped in coil and gives intense blue color. On heating, helical conformation is disrupted and it loses its capacity to bind with iodine. On cooling, the original conformation is recovered. Then Benedict's test. Take 5 ml of Benedict's reagent in a test tube. Add 8 drops of test solution. Mix well and boil for 2 minutes. Once again, Benedict's test. We will be performing Benedict test. Take 5 ml of Benedict's reagent in a test tube and add 8 drops of test solution. Mix well and boil it for 2 minutes. No colored precipitate. Starch is not a reducing sugar. Acid hydrolysis. Take 5 ml of test solution in a clean dry test tube and add 4 drops of concentrated H2SO4 and keep it in a boiling water bath for nearly 10 minutes. Once again, acid hydrolysis. Take 5 ml of test solution in a clean dry test tube and add 4 drops of concentrated H2SO4 and keep it in a boiling water bath for nearly 10 minutes. Cool under running water and then neutralize by adding 5 drops of 10% sodium carbonate till the effervescence ceases. The solution is hydrolyzed. Once again, take 5 ml of test solution in a clean dry test tube and add 4 drops of concentrated H2SO4. Don't add HCl. We are talking about H2SO4. And keep it in a boiling water bath for nearly 10 minutes. Cool under running water and then utilize by adding 5 drops of 10% sodium hydroxide, sodium carbonate till the effervescence ceases. Carbonate, don't confuse with reagent. Hydrolysis, we are performing hydrolysis of starch. So we will be adding concentrated H2SO4. Four drops of concentrated H2SO4 and water bath for 10 minutes. Then later sodium carbonate is added. Then Benedict test after hydrolysis. Take 5 ml of Benedict reagent in a test tube and add 8 drops of hydrolysate of test solution after hydrolysis. Mix it well and boil it for 2 minutes. Brick red precipitate hydrolysate contains reducing sugars. Unknown carbohydrate solution. Molish test. Reddish violet ring. Carbohydrate confirmed. Then iodine test. Deep blue color polysaccharide starch. Confirmatory test. Benedict test. No red precipitate. Benedict test after acid hydrolysis. for doing acid hydrolysis, we will be adding concentrated H2SO4 4 to 5 drops and then heat it. After cooling, add sodium carbonate. I have some doubts regarding this, so once again read that. Take 5 ml of test solution in a clean dry test tube and add 4 drops of concentrated H2SO4 and keep it in a boiling water bath for nearly 10 minutes. Cool under running water and then neutralize by adding 5 drops of 10% sodium carbonate till the effervescence ceases. Then no deep blue color, maybe mono or disaccharide. Benedict test, red precipitate reducing mono or disaccharide. No red precipitate, non-reducing sucrose. Sucrose confirmatory test, Benedict's test after acid hydrolysis, Selevanov's test. But what if the red precipitate is present? Reducing or mono disaccharide is there. Barfoit's test. Barfoit's test will create a red scum, reducing monosaccharide, glucose or fructose. No red scum, that means reducing disaccharide, maltose or lactose. Then later we will be performing Selevanov's test, which will indicate cherry red color in case of fructose, no cherry red color in case of glucose. Then we will be performing Osazon test, long needle shaped crystals, because that sample is nothing else but fructose. Then after performing Barfoit's test, if we got no red scum, we directly perform Osazon test, which will create either sunflower shaped crystals which indicate maltose or powder puff hedgehog shaped crystals or lactose. Then identification of unknown carbohydrate. First, we are going to perform Molish test. 2 1 ml of test solution, 2 to 3 drops of 1-alpha naphthol is added and mixed. Throughout the sides of the test tube, concentrated H2SO4 is added so as to form 2 layers. Appearance of reddish violet ring at the junction of the 2 liquids that indicate presence of carbohydrate. Then iodine test. To 1 ml of the given solution, 2 to 3 drops of iodine is added. No color, maybe mono or disaccharide. Then Benedict test. To 1 ml of solution, 3 ml of Benedict's reagent is added, mixed and boiled for 2 minutes over a small flame. Red precipitate may be reducing sugar. That means we don't need to perform the hydrolysis, acid hydrolysis. Barfoit's test. To 2 ml of given sample, 2 ml of Barfoit's reagent is added. Keep the test tube for 3 minutes in a boiling water bath. Cool under running water. Phosphomolybdic acid is added drop by drop with mixing till the solution becomes clear. Intense blue color may be monosaccharide. That is Barfoit test. Phosphomolybdic acid is a requirement. Phosphomolybdic acid. Barfoit's test, Barfoit solution, our solution, heat, cool, then phosphomolybdic acid is added. Selevanov's test. To what was the result? Intense blue color was there. Barfoit's test. Selevanov's test. To 3 ml of Selevanov's reagent, 1 ml of given sample is added. Boil for 30 minutes and cooled rapidly. A red color is obtained. Presence of fructose confirmed. Selevanov's test. Barfoit test positive means maybe monosaccharide. Selevanov's test positive means fructose. Result: the given unknown sample contains fructose. Never forget to write that down. Then some other aspects related to biuret test. Biuret test is a test for protein. So learn that. Biuret test. Cupric ion in an alkaline medium forms a violet colored complex with peptide bound nitrogen of proteins. It is a general test for proteins. The minimum requirement for a positive test is the presence of 2 peptide bonds, dipeptide, in the molecule. Individual amino acids and dipeptides do not answer the test. 2 peptide bonds are necessary. So 1 peptide bond will not count. 2 is the must. 2 is a must. So dipeptide do not answer this test. Addition of excess amount of copper sulfate should be avoided as blue color may develop instead of violet color. Sulfur test for cysteine and cysteine. When cysteine or cysteine protein containing these amino acids are boiled with strong alkali, organic sulfur is converted to inorganic sulfide. Addition of lead acetate to solution forms brown or black colored lead sulfide. So methionine do not answer the test as the sulfur in methionine is not released by alkali as it is in the thiostate linkage. Methionine do not answer the test as the sulfur in methionine is not released by alkali as it is in the thiostate stage. Molish test for carbohydrate. Protein containing carbohydrates as prosthetic group like glycoproteins answer this test. Perform Molish test with albumin solution, not the formation of a violet ring at the junction of two liquids. That's why while performing this unknown test identification, first test is not Molish test but Biuret test. Okay. The Newman's test. Newman's test for organic phosphorus. The organic phosphate is phosphoproteins is converted to inorganic phosphate on boiling with strong sodium hydroxide solution. The organic phosphate is the phosphoproteins is converted to inorganic phosphate on boiling with strong sodium hydroxide solution. Inorganic phosphate reacts with ammonium molybdate in the presence of nitric acid to form ammonium phosphomolybdate precipitate which is candy yellow in color. Then xanthoproteic test. Aromatic rings in tyrosine and tryptophan undergo nitration on treating with concentrated HNO3 to form nitro derivatives which are yellow in color. In an alkaline medium, yellow color turns orange due to ionization of phenylalanine alone does not give any positive result as it does not undergo nitration under these conditions normally. The nitrates in the basis of yellow staining of the skin in contact with the concentrated nitric acid. So aromatic rings in tyrosine and tryptophan undergo nitration on heating with concentrated HNO3. Xanthoproteic test. Aromatic rings in tyrosine and tryptophan undergo nitration in treatment with concentrated HNO3 to form nitro derivatives which are yellow in color. In an alkaline medium, yellow color turns orange due to ionization of phenylalanine alone does not give positive result as it does not undergo nitration under these conditions. Normally, this nitration is the basis of yellow staining in the stain in contact with concentrated nitric acid. Then isoelectric precipitation is a test in case of protein. Proteins have minimum solubility at their isoelectric pH. Minimum solubility in the sense they just precipitate out. No solubility. So isoelectric pH, during isoelectric pH, proteins have minimum solubility. Proteins are easily precipitated from their solutions by adjusting. Proteins have minimum solubility at their isoelectric pH. Proteins are easily precipitated from their solutions by adjusting pH close to their isoelectric pH. This is achieved by addition of an acid and an alkali. pH range of BCG is 3.8 to 5.4. When more acid is added, the pH changes and flocculent precipitate formed by casein at its isoelectric pH resolves curdling of the milk is one example of isoelectric precipitation. Casein forms curd and then milk is curdled. This is because lactic acid produced by the fermentation process lowers the pH to isoelectric pH of casein, thus precipitating casein from milk. Test for albumin. Albumin. From where we could get albumin? From egg albumin, right? Albumin flakes developed from egg albumin or something. So Biuret test is the primary test. To 1 ml of test solution, add 1 ml of 5% NaOH and 1 to 2 drops of 1% CuSO4. Next, well, once again I will be reading. Add to 1 ml of test solution, add 1 ml of 5% NaOH solution and add 1 to 2 drops of 1% CuSO4. Mix well. Violet color is formed. Presence of protein. Then isoelectric precipitation. Take 3 ml of test solution. Add 2 drops of bromocresol green or BCG indicator and mix. Add 1% acetic acid drop by drop until a green color is formed, which indicate the pH is 4.6. To 1 ml of test solution, add 1 ml of 5% NaOH and 1 to 2 drops of 1% CuSO4. Mix well. No need to heat. Violet color is formed. Presence of protein. Biuret reagent, or either you can perform biuret test by just adding that biuret reagent. Already mixed solution is also available. Just introduce that biuret reagent. We will be learning this procedure after some time. Okay. Then isoelectric precipitation. Take 3 ml of test solution, add 2 drops of bromocresol green BCG indicator and mix, and add 1% acetic acid drop by drop until a green color is formed, which indicates the pH is 4.6. Isoelectric precipitation. Take 3 ml of test solution, add 2 drops of bromocresol green indicator and mix, and add 1% acetic acid drop by drop until a green color is formed, which indicates the pH is 4.6. Isoelectric pH 4.6. Bromocresol green, then acetic acid, 1% acetic acid. Okay. No Kurdi precipitate. Presence of protein. Maybe albumin, gelatin or peptone. Absence of Kurdi precipitate indicate the presence of albumin. Precipitation by heat and acetic acid. Till 3/4 of the test tube, add test tube solution. Hold it over a flame in a standing position and boil the upper 1/3 of test tube. The lower portion serves as control. To this, add 1 to 2 drops of 1% CH3COOH. Coagulation occurs. Presence of albumin. How to do this? Till 3/4 test tube with the test tube solution, then hold it over a flame in a standing position and boil it over 1/3 of test tube. The lower portion serves as a control, and add 1 to 2 drops of 1% CH3COOH. Coagulation occurs. CH3COOH, acetic acid. Presence of albumin. Sulfur test. To 2 ml of test solution, add equal volume of 40% NaOH and boil for a minute. Cool and add 2 to 3 drops of lead acetate. Mix well. Brown color is obtained. Presence of albumin. Sulfur test. This is a new test. How to perform this? 2 ml of test solution. Add equal volume of 40% NaOH. Add equal volume of 40% NaOH into this solution and then boil. Later add lead acetate. That will create a brown color is formed. Presence of albumin. Sulfur test. To sample, add equal amount of NaOH, 40% NaOH, and boil for a minute. Cool and add 2 to 3 drops of lead acetate. Mix it well. 40% NaOH, heat, then cool. Along with that, add lead acetate, 2 to 3 drops. What will be the result? Brown color is obtained. Presence of albumin. Molish test. To 1 ml of the test solution, add 2 drops of Molish reagent. Mix it well. Add 1 to 2 drops of concentrated H2SO4 carefully along the sides of the test tube. Brownish violet ring at the junction of two liquids. Presence of albumin. Albumin is a glycoprotein. Then precipitation by neutral salts. To 2 ml of test solution, add solid ammonium sulfate and mix until the solution is saturated. This stands for 5 minutes. White precipitate is formed. Albumin is precipitated by full saturation with ammonium sulfate. ammonium sulfate. We just complete the test for albumin. Once again, let's learn that. Test for albumin. Earlier we mentioned some common tests. Okay. Test for albumin. First, biuret test. Biuret reagent, heat, no heat, just add biuret reagent, or either add 1 ml of test solution, add 1 ml of 5% NaOH and 1 to 2 drops of 1% CuSO4, mix well, violet color is formed, presence of protein. Then isoelectric precipitation. Isoelectric precipitation. Take 3 ml of test solution, add 2 drops of bromocresol green indicator and mix and add 1% acetic acid drop by drop. A green color is formed which indicate pH is 4.6. And no Kerdi precipitate indicate presence of albumin. Presence of protein may be albumin, gelatin or peptone. Absence of Kerdi precipitate indicate the presence of albumin. Absence of Kerdi precipitate. Then precipitation by heat and acetic acid. Till 3/4 of the test tube with the test tube solution. Hold it over in a flame in a standing position and boil the upper 1/3 of test tube and lower portion serves as a control. To this add 1 to 2 drops of 1% CH3COOH. Coagulation occurs, presence of albumin. Then sulfur test. To 2 ml of this solution, add equal volume of 40% NaOH and boil for a minute. Cool and add 2 to 3 drops of lead acetate. Mix it well. Brown precipitate. Brown color is obtained, presence of albumin. And Molish test is performed. That was turned out to be positive. Then precipitation by neutral salts. Which neutral salt we are using? Ammonium sulfate. Into 2 ml of test solution, add a solid ammonium sulfate and mix until the solution is saturated. Let this stand for 5 minutes. White precipitate is formed. Albumin is precipitated by full saturation with ammonium sulfate. Then test for casein. Experiment biuret test. Biuret test is performed, turned out to be positive. An isoelectric precipitation. Take 3 ml of test solution, add 2 drops of bromocresol green indicator and mix and add 1% acetic acid drop by drop until a green color is obtained which indicate pH is close to 4.6. A Kerdi precipitate is formed. Casein may be present. Casein is precipitated at its isoelectric pH 4.6. If this precipitate is not there, that means that will be albumin. Okay. Then Neumann's test. In case of albumin, we performed sulfur test. How to perform sulfur test? To solution, add 2 ml solution, add equal amount of 40% NaOH, heat it and later add 1 to 2 drops of lead acetate. Then in case of this albumin, we performed sulfur test. But here in case of casein, we will be performing a new test called Neumann's test. To 5 ml of test solution, add 0.5 ml of 40% NaOH, heat strongly, cool under tap water, add 0.5 ml concentrated HNO3, filter the precipitate to the filtrate, add a pinch of ammonium molybdate and warm gently. Canary yellow precipitate is formed. Presence of casein. Casein is phosphorated. Once again, I will be repeating. To 5 ml of test solution, add 0.5 ml of 40% NaOH, heat strongly, cool under tap water, add 0.5 ml concentrated HNO3, filter the precipitate to the filtrate, add a pinch of ammonium molybdate and warm gently. Ammonium molybdate. Canary yellow precipitate is formed. Presence of casein. Casein is phosphorylated. And precipitation by neutral salts. Take 2 ml of test solution, add equal volume of saturated ammonium sulfate solution, mix and allow to stand for 5 minutes. White precipitate is obtained. Presence of casein. Casein is precipitated by half saturation with ammonium sulfate. ഹാഫ് സാച്ചുറേഷനിൽ തന്നെ precipitate ചെയ്യുന്നതാണ് കേസൻ. സോ കേസൻ is precipitated by half saturation by ammonium sulfate. Okay. Then sulfur test. To 2 ml of test solution, add equal volume of 40